All right, good afternoon. I am going to set this part of the lecture off just a little bit. Again, this is still testable material, but we want to take more of a liberal arts view of things. Because we have mentioned two women's names already in class, and next Monday we're going to mention two more, Dorothy Hodgkin and Rosalind Franklin. You may already have heard of Rosalind Franklin. We've got a little more information to tell you about her. But we've already, why do I keep mentioning women's names when I talk about protein chemists? And the reason is the field was actually more open to them. I want to tell you about their stories, and then I want you to think about why you think that this pattern is definitely not just cherry picking. This is actually a major pattern, and I'm going to show you that in a second. So first of all, you've already seen these cartoon structures. You've seen how important they are to the field. And these were actually developed by this woman, Jane Richardson, whose husband was a crystallographer, David Richardson. Jane worked with her husband, and her husband found out where all the atoms were, and he did a lot of structures that people still use today. But more people, everyone in the field, uses Jane Richardson's depictions of what the structure means. Because what she did, remember, there's so many atoms, you've got to be able to boil it down somehow. And it's all, just looks like a spider web of circles if you don't use your brain to make it simpler, to say what is important and what is not important. And that is actually a very hard thing to do sometimes. Not only do you show what's important and not what's important, but make it in a sense beautiful as well. And that's what Jane Richardson did. There's a whole story about this, and this is in one of the main journals of the field called Journal of Biological Chemistry. And it talks about, for example, this was a hand-drawn schematic of copper zinc superoxide dismutase. You can see how this immediately communicates where are the two metals, what is the overall structure of the protein. Now there are some things you don't see. You don't see exactly where the active site is, but let me give you a hint, it's right where those two metals are. So you can't see the side chains, but the point is you get a feeling immediately for what this protein is, and that can be related to how it works. And Jane got this because she had a philosophy background. So normally when they're looking for like biochemistry jobs, they don't advertise for philosophers. And yet she was, so for example, one of the first examples was published in the early 80s. And Jane was looking at the recurring patterns of motifs and realized, oh, somebody should do this artistically to show these motifs are important, that there are folds, and the folds can make several proteins that look kind of different. You can see how they're the same if they have the same secondary structure and tertiary structure. And so there had only been 75 protein structures solved at the time, but that was enough for them to see the patterns and, for example, to hand draw it. Based on her hand drawings, now we have that setting in pi-mole that it can do it for any protein structure. We give it the atoms, it gives us the cartoon. So you can see the pencil smudges on this. Jane was a good artist, and she also had the philosophy helped her identify what is really important and how do I depict this in a way that shows what's important. Another thing that's important here is the direction of the chain. The N to C direction is shown by the arrows very clearly, and that is a big part of what Jane contributed to this whole field. So you have these early pictures, and then I showed you these pictures before. We don't have to draw them anymore. We can let the computers draw them, but we're still using Jane's sense of style. And we went through these to show you how originally, just look at the difference between the 1961 ball and stick and the 1981 ribbons. Now, in the lab, as you work on pi-mole, you'll be able to turn things on and off, and I want you to think of things like Jane Richardson did. I want you to say what's really important, and I want to leave off the stuff that's not important and show you everything of what is actually important. So you want to be a little bit of a philosopher like Jane. You have to look at the meaning of the thing and find the one piece of meaning that unifies what we're talking about, rather than the bunch of disparate things that don't appear to have any meaning at all. Just want to show you now we can expand this, and there are people talking about modeling an entire cell atom by atom this way. And when we look at these things, now we have more than 75 like Richardson did when she started. But we can still name proteins certain ways. And the last thing I want to mention is what are superfolds or superfamilies. It's the level above the fold. When you looked at Jane Richardson's picture of superoxide dismutase, you saw what its fold was. And there are also ways to classify things as above the level of folds. For example, myoglobin has all alpha helices, basically, like 80%, right? So what other things that don't have the same fold as myoglobin, but other things that are all alpha helices, we can make that into a superfamily, and we can say it's the all alpha superfamily. And these might have some things in common. They'll definitely structurally have some structural aspects in common. And so we can talk about the all alpha superfamily. And you can see here's 4 examples of it of various sizes, but these are all mostly all alpha, and they have very different arrangements of the alphas, but we can talk about what they have in similar things and what they have differently. Then if you can have an all alpha superfamily, you can also have an all beta superfamily. Here it is, and in fact the immunoglobulin sandwich, I mentioned that in class, is here on the right. It's an example of an all beta superfamily member. You can arrange the beta sheets in sort of a propeller. You can have them in a beta helix, but this is not like an alpha helix. This is a true beta sheet. It's like a 3-sided beta sheet put together, and that is a member of the all beta superfamily. Then there's a family, there's 2 other ones. What do you do when it has both alpha and beta? Well, there's 2 different things, and I want you to look at these and try to figure out the difference between them. Here's 3 proteins in the alpha/beta superfamily, and here's 3 proteins in the alpha plus beta superfamily. So look at these, and we're going to go back to the last one. I want you to think about, why do we put these in one group where we say it's plus, and we put these in one group where we say it's a slash? What could that mean? So I'll give you the answer. Pause it if you want to think about it a little more. But the answer is, if you look at these, the 2 areas are more integrated. It's not like you have a beta side and an alpha side. In fact, you have, in one of them, you have like a beta core, and then you have all these alphas around it. They're mixed up more. And so you put a slash to show that they're sort of mixed up together. Alpha plus beta, on the other hand, you have sort of an alpha side and a beta side. All the alphas are on one half of the protein, all the betas are on another. This one is even a beta barrel, but it has a little alpha part up here. So apparently by their mode of classification, they said this is enough alpha in it. I'm not going to call it an all beta, I'm going to call it an alpha plus beta. And so we're going to return to this, but this is going to be in your homework. There is a computer program that, again, I showed you the computer program where the computer was given structures and said find motifs in these. Here's a computer program where the computer was given the whole structure and said to find superfolds. What it did is it took all the structures, and this is, I don't know how many structures, like 1,000, and it found ones that had structural tertiary structure in common. And it put those together. It's called the galaxy of folds, and it put those together in a group if it saw similarities between the tertiary structure. Now, it did not know anything about secondary structure. Everything that comes out of here is just from the computer objectively looking at tertiary structure, putting similar tertiary structures together. But the question is, what did the computer see? And it saw these different areas, and I want you to see down here, I've highlighted, the different colors mean things. What we did is the computer put the different, the things that it said was more similar, it put those proteins closer, it put them all on a table, and it slid the ones that it looked like they were more similar to each other, it slid them closer together on the table. Then, only then, at the end of its whole thing where it sort of put the things it thought were close together. So if it's close together, the computer thought it was close together. Then we turned on the colors. We colored them according to the human categories, where if the humans had said that it was all alpha, we would color it blue. If the humans said it was all beta, we'd color it cyan. And look at this. The computer and the humans agree. The all alphas are put together in one group over here, and the all betas are in a completely opposite group on the other side of the galaxy. And it becomes really interesting. That's like confirming what's obvious. It gets really interesting when you compare alpha slash beta, which is red, down here, alpha plus beta, which is yellow, and that's in the middle. Notice that those are in different spaces. The red is right next to the dark blue. It's far away from the light blue. I want you to think about what that means. Then you have, what does it mean? And I have homework questions for you about this. So you have orange, which is multi-domain proteins. Those are sort of scattered all around. And you have membrane proteins, which is magenta. And just see there's a little membrane protein magenta spot here, and there's one over here. The last thing I want you to see is small proteins are colored green, and they are located up here. Don't worry if you can't see this very well. I will actually copy this and have this as a handout in a future class. But you can actually go ahead and start thinking about this right now, because the homework questions that involve this can already be answered on the basis of this. Okay, so that's Jane Richardson's contribution. Jane's work allowed humans to look at the proteins and to make these categories more effectively, because Jane highlighted what was really important. Yeah, and the main thing is domains from the same class, same color, come to lie in similar regions of the galaxy, which means the computer said they are more structurally similar. Okay. Here is a list, and this is what I want to talk about with women in science, because we've talked about 3 of them so far, or 2 of them so far, and we're going to talk about 2 more on Monday. 2 of 24 of them are crystallographers, and I noticed when I saw this, this is cut off a little bit. There are actually, there's another row of 6. But I thought it was really interesting that we talk about 2 of them on the same day. We have Rosalind Franklin up here, and we have Dorothy Hodgkin over here. And you can see Rosalind has DNA. Dorothy has this weird kind of organic chemistry cofactor. We'll talk about that. But the other ones are from all sorts of different areas. Sally Ride was an astronaut. Marie Curie was a nuclear chemist. Ada Lovelace was a computer scientist. You have lots of different people and different things. You can see there's a lot of them that were involved in the space program. But there's a lot of them that were involved. It's a pretty small field, so it's weird to have 2 of 24 mentioned as being specifically X-ray crystallographers. We'll talk about the technique on Monday, and we'll talk a little bit more about what Rosalind Franklin and Hodgkin did. But I want to prepare you a little bit, because X-ray crystallography identifies where all the atoms in something are in 3D space. It's easy to do for small molecules, and of course they did those first, and then they worked their way up to bigger molecules, and you can even do proteins that way now. Look what Hodgkin did. Not just one important structure, but 3. Penicillin, kind of an important molecule. Vitamin B12, which is in the middle, in 1955, Hodgkin found this structure. And in 1969, Hodgkin first grew crystals of insulin. We talked about how important insulin was to medicine, how important it still is to biotech. And here we have, it was 34 years between when they first got the crystals of it and when they were actually able to determine its structure. And so Dorothy Hodgkin also, after she accomplished all these things, she was always a person who was involved in many fields. For example, she actually had a role to play in politics. There's a whole book about, during the Cold War, scientists had a role to play in how the Cold War unfolded politically. And Hodgkin was one of the main people. Because of her reputation, she was able to cross borders. She was able to talk to people in the Soviet scientists. And there were political aspects of science. Scientists talking to scientists had political ramifications that some people argue were were influenced by politics, and there were politicians trying to influence science, but also could science have influenced politics in a certain way? Really fascinating stuff. I bet you didn't expect poli-sci to be one of our contents, but I'll let the political scientists talk about what parts of this were good and what parts of this were bad. I'm just going to say Hodgkin was actually involved in this, and there are scientists who have, for example, won Nobel Peace Prizes because of their work in politics. But when you talk about crystallography and the history, you can't help but notice that Dorothy was the first name of a prominent crystallographer, and Rosalind was another one. Rosalind Franklin used crystallography to determine the double helical structure of DNA. All of these, Hodgkin was definitely, clearly, undoubtedly central to all the things that she did, and she was in the field for a long time. Rosalind actually died young as a result of the radiation that she exposed herself to as part of her work. So Rosalind Franklin actually has the one major discovery, whereas Hodgkin has many other discoveries in a row. But they were both big contributors to the science of this one little area, this one lecture in biochemistry. And so there's a question, there's articles about this. You can read the whole article, but here's a quote from it. The collaborative ethos, and these were part of teams. We're going to talk about the team that Franklin was involved with was a little dysfunctional, with people taking credit for what other people had done. We're going to talk about that, and it's really complicated. But it takes a lot of people to determine an X-ray structure. The science itself involves geology, biology, chemistry, radiation physics, virology, immunology. All these things are things that you have to know a little bit about. And so people who are brought up in the traditional settings of academia, traditional disciplines, were not necessarily well prepared to go outside of their discipline. I want you to think about why that would affect the gender balance of a field. And of course, this is historical, the gender balance in the 20th century. But it's important for us to know this history. Here's a portrait of Hodgkin, and you can see, I believe that's the insulin structure she has on her desk. They used to literally piece those together with balls and sticks, and now we can just fire up PyMol and you can do it on your laptop. I think this is a nice depiction. She had a very long career and an illustrious career. She is one of the great scientists. And one of the things about this is that she had like Jane Richardson had a philosophy background, Dorothy Crowfoot Hodgkin had a archaeology background. As a teenager, she would work with her parents' archaeological digs. I believe both her parents were archaeologists. And she would draw the tessellations, the different tiles that they had. It turns out later, when she was growing protein crystals and they were determining the structure of those crystals, crystals come together exactly like a mosaic does. Symmetry operations are involved in each. So the two-dimensional order in a plane has restraints and there's symmetry that determines mathematically how they relate, and that symmetry is crucial to why it is that we can get good structures from a crystal when we can't just get them from a jumble of proteins. The thing about this is Hodgkin, of course, insulin took decades to figure out its structure of. So what they did is they would grow the crystals, they would shoot it with x-rays, they would try to find out where the patterns and symmetry were. Where are the atoms? You can't necessarily see the atoms at first, but you can see the blobs of atoms, and you can see how those are arranged. And when they did it with insulin, they pretty quickly saw that they saw hexagons over and over again. And it turns out that insulin has two chains, and the two chains are in a trimer. 2 times 3 equals 6. Insulin does have a hexagon of alpha helices that are arranged in its structure. For the early pictures of insulin, they look like hexagons, and Dorothy Hodgkin drew them like this. Because she had some artistic ability, she could make them beautiful. And if you look at that picture on the left, that is a scientific depiction of where the helices in insulin are at low resolution. And also in the middle, there's a metal holding it all together. It looks like a flower, but it's actually an insulin structure. And so Hodgkin realized this, and she had friends who were outside the discipline, and they were talking one day, and I don't know who had the idea, but they got the idea of, this is a pretty design. Let's take this and try to adapt it for other kinds of art. So the artist put this on wallpaper. This is actually an adaptation of an insulin structure at low resolution that the artist then played with and simplified and turned into art. Here's one that's more simplified. You can see all 6 helices, you can see the metal in the middle, but unless somebody tells you, you don't know this is insulin. But insulin does have this hexameric structure, and that's reflected in this art. There's a whole exhibit about this, and I was actually wandering in London. I highly recommend just wandering around in London and finding walking in through open doors. There is a museum by the Wellcome Trust that funds a lot of science, and it was about from atoms to patterns. It's how I found out that Hodgkin had these friends who were artists who made wallpaper and other art out of early patterns of different proteins. So here's some examples, and you notice this one is not hexameric. I believe this one is a myoglobin or some kind of globin structure, and the same kind of things were done for the globins. There was an exhibition of science in 1951. A lot of these artworks were exhibited there, and you can even see how there's kind of, you can even see a little bit of what the exhibition looked like and how the art and the science were sort of integrated. I think those light fixtures are really cool, some kind of fiber or crystal or something. And, 1951, they did a lot of science and art like this. Is that a disco ball up in there? I'm really not sure. I do know that at the time, they put the insulin wallpaper up in the restaurant on the South Bank of London. And here's a picture of the dining room, low resolution, but you can sort of see the hexameric pattern in the back. They have curtains that had the pattern, and that was part of the exhibition. You could dine under these protein structures that were turned into art. And there's even a set of plates. I would love to find these plates somewhere somehow. I have never looked for them, but I've just realized the internet's working. Maybe I should try. These are different protein structures, but I believe that the top ones at least are related to insulin. Then artists take liberties, and you choose how you color them. You choose in Pymol how you color the Pymol stuff. And there was even a crystal of beryl, which is a mineral, and crystallography was first developed by geologists that were looking at mineral crystal structures. And it turns out that beryl 8-9 has a nice little pattern to it that was turned into an evening dress that was worn by Lady Alice Bragg at the International Union of Crystallography meeting in 1951. So Nobel Prize winning husband, apparently you can make people will make your crystal structures into a dress pattern for you. And then finally there's hemoglobin lace, which, and that does show you the earlier structures. I believe those were some form of globin, and it had the heme in the middle. Because the iron is a very dense atom relative to carbon, nitrogen, oxygen, right? So if you're looking for electrons, you'll see the iron light up like a beacon. And so this was an early structure of hemoglobin, and they made lace out of it. I think that's kind of ironic because hemoglobin is blood. We think of that as being very different from fancy lace. Here they are put together. That's an artistic statement. And you can do the same thing. You can put things together like they did. I want to encourage you to think in an interdisciplinary way. Talk to your friends from other disciplines. And it's actually surprisingly easy for somebody who's put a lot of work into learning science to make connections to other fields if you just open yourself to it. So, with all this around, why are there so many women scientists in the mid-20th century? Why were they protein chemists? Technically DNA chemists, but they were X-ray crystallographers and things like that. Why is biochemistry inherently more open? It is an interface between other established fields. I want you to think about how that could play out. And then, where are the other interfaces today? Where are the interfaces in your own personal life where you can put things together and see things that somebody else doesn't see? Not by creating a whole new world, but by taking the world we live in and just looking around and putting things together with your own mind. I hope to teach you how to put things together in biochemistry terms, but I want you to think bigger than that as well. So that is my liberal arts lecture. I hope I tried to put together some things like that for you. Have a good weekend, and we will come to class on Monday for a special light show that will teach you X-ray crystallography and what Rosalind Franklin did to earn her name in science. All right, see you then.